Hi, my name is Mark Mattson. I'm the host of this new series of videos called Brain Ponderings. These are going to be on YouTube, but also in podcast format audio on Spotify and Apple audio. What these are going to be is conversations between me and experts in the fields of brain research and neurology. And the idea is to present the information at a level understandable by most people, including non-neuroscientists with an interest in the brain, how it got to be the way it is, how it develops from the fertilized egg to the newborn and adult, how it changes during adult life, and what's going on in the brain, in the nerve cells, other cells. That explains things like learning and memory, emotions, anxiety, fear. We're going to talk about some of the effects of life's challenges on the brain. So, for example, intellectual challenges, exercise, fasting, stress, bad stress, chronic psychological stress. We're going to talk about how the brain affects the body and vice versa. And then I'll have a number of experts on certain neurological disorders, ranging from psychiatric disorders to Alzheimer's, Parkinson's disease, stroke, and others. We'll talk about developmental brain disorders such as autism spectrum disorders. We'll have multiple talks about drugs used in clinical practice, drugs of abuse, and some of the promising drugs on the horizon, for example, psychedelics, which are increasingly demonstrated to be effective in reducing depression and perhaps even in overcoming addiction. And we're going to talk also about ethics in neuroscience research. As the research progresses and it becomes able to modify the brain through using molecular genetic tools or other approaches, for example, brain stimulation, it becomes important to debate, how much intervention is ethically reasonable. Okay, a little bit about myself. I'm, of course, a neuroscientist. And my background is I grew up in southern Minnesota, Rochester in particular. And we lived on a farm, and my father and I and my brother trained and raced harness horses, standardbred horses, trotters, and pacers. And we'd race at county fairs, sometimes go down to Kentucky if we had a good horse that we'd entered in stake races. And so I wanted to be a veterinarian. So I went to Iowa State University in Ames, and I was in pre-veterinary medicine. And at that time in the 1970s, you could apply for veterinary medicine school after your second year of undergraduate learning. And I did, and I didn't get in. My grade point wasn't quite high enough. It's very competitive. That was disappointing, but I became interested in research, how cells work, how organs work, how the body and brain work. So I decided to pursue graduate programs and get first a master's degree and then a PhD. I went to Texas, to North Texas State University, and studied how a hormone released from the brain that stimulates the adrenal glands to produce the stress hormone cortisol, how that system works, how that signal from the brain leads to increased production of cortisol. Then, and I met my wife, Joanne, when I was in Texas. Then I started a PhD program at University of Iowa. And when I was at Iowa, my wife was a med tech at the University Hospital. In Iowa, I studied a neuroendocrine system that controls molting in crabs. And after my PhD work, I was recruited to work with Stan Cater at Colorado State University in Fort Collins. And it's there that my interest in basic neuroscience really took off. I studied how nerve cells grow and form connections in the developing brain. And my main discovery there in that postdoctoral research was that the most abundant and important neurotransmitter in the brain, an amino acid called glutamate, controls the growth and formation of synapses in the developing brain. And after that, I went to the University of Kentucky, and it was during this time I became interested in what goes wrong in brain aging and neurodegenerative disorders such as Alzheimer's. And so we did a lot of research at Kentucky, elucidating what's going on at the cellular molecular levels in Alzheimer's, Parkinson's disease, and stroke, as well as epilepsy. And also when I was at University of Kentucky, we started doing experiments to look at how intermittent fasting affects the brain. I was then recruited to the National Institute on Aging, which is part of the NIH. The laboratories at the National Institute on Aging are located in Baltimore on one of Johns Hopkins campuses. And so I was there 20 years, from 2000 to 2020, essentially. And so there, did a lot of work, and maybe some of that we'll talk about with some of my guests, as in some cases it's related to what they're doing. Okay, so that's me in a nutshell. Now what I want to do is present a little bit of information about the brain to kind of introduce the general areas of consideration we'll talk about in these Brain Ponderings podcasts and videos. This is a human brain here, looking from the bottom, the top, the right, the left. The human brain has about 100 billion neurons and about 100 trillion synapses, the connections between the neurons. So that means there's approximately 1,000, each neuron has approximately 1,000 synapses. There are also at least as many cells in the brain that are not neurons. And these cells are called glial cells. And we'll talk a little bit about their functions, which turn out to be quite important. Now, the brain is remarkable in many respects. Its capabilities in terms of learning and memory, creativity, imagination, and so on. And it's also highly energy efficient. This is a figure from an article in Scientific American 10 years ago, comparing the information storage, processing speed, and energy efficiency of what was at that time the largest, most powerful supercomputer, the human brain as well as the cat brain, iPad 2. And the bottom line here, of course, now there's more powerful supercomputers, but at this time, the human brain was maybe about 10-fold less storage capacity as the largest supercomputer. It had somewhat less slower processing speed, but it was 500,000-fold more energy efficient than the computer. So that's pretty interesting. How is it that these cells process information in a highly energy efficient manner? And of course, the explanation to that, as to all explanations with regard to the brain or anything in biology, has to be explainable in terms of evolution. What were the advantages of being highly energy efficient? Well, the obvious one is you don't have to consume as much food to keep your brain and body running. We're going to talk about brain evolution, human brain evolution, but also lower animals. Here's some brains from different animals: fish, frog, snake, pigeon, rabbit. And as we go up the evolutionary scale, up the branches of the evolutionary tree, brains tend to get bigger, and particularly what the cerebral cortex, which is involved in higher functions, learning and memory, creativity, imagination, decision-making is very important. We study the brain in animals, mainly mice and rats, but also insects like the fruit fly and other organisms which have very few neurons. The neurons here are shown in green. This is the eyes, two big lobes of the brain and neurons. These are mammals, mouse brain, two different non-human primate brains, a rhesus macaque, chimpanzee, and the human brain. And we'll talk about what's different in terms of the structure and function of the human brain. But one brain region that increased a lot from chimpanzee to humans is it's called the prefrontal cortex at the front of the brain. And this brain region is very important in decision making, which turns out humans are very good at. Here's another perspective on brain evolution. Food scarcity was the major driver, if you will, of brain evolution, such that individuals whose brains, as well as bodies, functioned well in a food-deprived state were the individuals that survived and passed their genes on. And we've done a lot of work in animals and some in humans on how eating patterns that involve extended periods without food may benefit the brain, and how the brain responds in positive ways, improvements in cognition, for example, even mood. Again, from an evolutionary perspective, all the tools invented by humans that we know of, the early ones, had to do with acquiring and processing food. And as our ability to invent tools to pass information on between individuals and across generations through language, the size of the brain increased. Okay, we'll talk about brain development. We'll talk about development in the womb and how the mother's lifestyle and experiences during pregnancy may affect the development of the brains of their baby. We can study developing brains in some ways best in animals, where we can actually go in and take out brains at different ages and study them, or take the brains out from a developing in the rat embryo and dissociate or separate all the cells, put them in culture. This is an individual nerve cell that I put in culture about in the 80s. This is from a fetal rat, a pregnant female rat. And when you put these cells in culture, initially the cell is a sphere, kind of like the spheres of blood cells, but then these cells will attach to the bottom of the dish and they'll grow a long process, which is the axon, and several short processes, which are dendrites. And these cells will form connections with other cells in the dish. So that's one way we can actually study the growth factors that affect the growth of dendrites and axons, and factors that affect and mechanisms that affect the formation and maintenance of synapses. And then in the adult brain, the brain can function very well till late in life. There's many people who live into their 90s and their brain is functioning very well. They're learning and memory, for example. On the other hand, unfortunately, there are many people whose brain does not age gracefully, if you will, or successfully, and they may develop dysfunction and degeneration of nerve cells, such as occurs in Alzheimer's disease. Now, we can study brains in living humans, for example, with magnetic resonance imaging, MRI. We can study animals in very highly specific ways. For example, this is a mouse in which a fiber optics light fiber has been put into the brain. This mouse has been genetically engineered so that when light is shined on nerve cells in its brains, the light can either activate or inhibit those nerve cells, depending on the specific genetic manipulation. And this allows one to, as the investigator wants, to activate or inhibit specific nerve cells and see how it affects the animal's behavior. Now, if I were to ask many people to name a neurotransmitter, the most common answers I get are dopamine or serotonin. And that's because they hear of dopamine for its role in addiction and pleasure, reward, or they hear serotonin for its role in depression. But it turns out that the most abundant by far and most important neurons in the brain deploy the neurotransmitter glutamate. Glutamate is the main excitatory neurotransmitter in the brain, and approximately 90% of the neurons, 90 billion neurons, are glutamatergic. Most of the other neurons use the neurotransmitter GABA. This is the inhibitory neurotransmitter. The other neurotransmitters, dopamine, serotonin, acetylcholine, norepinephrine, there are actually relatively few neurons that use these neurotransmitters. And the only way that these neurotransmitters affect behavior, mood, learning and memory, addiction is by modifying the activity in these glutamatergic neurons. Actually, I'm writing a book on glutamate, the brain's most important neurotransmitter, because it turns out that abnormal hyperexcitability of glutamatergic neurons is involved in epilepsy, which is fairly obvious, but also in Alzheimer's, Parkinson's, even stroke. Now, if you look at an individual synapse, so this is an axon here, and it forms a synapse on the dendrite of another neuron. And when one stimulates this axon, what happens is that in the case of a glutamatergic neuron, glutamate is released from the axon terminal into what we call the synaptic cleft. And then glutamate activates receptors that cause calcium influx into the dendrite. And it turns out this process is the process that underlies learning and memory. And we'll talk about that. So we have neural circuits formed during brain development. Turns out that the structure of the brain is constantly changing in relatively subtle ways, but in ways that we can actually measure and demonstrate. For example, there can be an increase in the number of synapses. This can be stimulated by things like engaging in intellectual challenges, by physical exercise. There can be an increase in the size of individual synapses. That's been demonstrated to be associated with learning and memory. Synapses can be removed, which is actually a normal process, but it can also occur in an unregulated manner in certain diseases. And then in neurodegenerative diseases, synapses can degenerate and neurons die. I mentioned that the brain contains many cells that are not nerve cells. These are called glial cells. There are 3 types of glial cells: astrocytes, oligodendrocytes, and microglia. The astrocytes play a very important role in energy metabolism, and they actually couple or regulate blood flow in response to activity in neurons. So if neurons are active, they send a signal to the astrocytes, and then the astrocytes send a signal to the blood vessel, which causes them to dilate, get bigger, and that results in more blood flow and more nutrient supply to the active neurons, which need that extra energy to keep functioning. The oligodendrocytes are the insulating cells. They wrap around axons and form these what are called nodes. And so this actually enhances the speed of conduction of the signal along the axon. It's like insulating a wire, metal wire. And then microglia are an immune cell in the brain. They move about the brain looking for bacteria, viruses, or even dying cells, and remove them. However, these microglia can cause inflammation in the brain, and we think that occurs in a chronic manner in Alzheimer's and even Parkinson's disease. The brain is very complex, obviously, with 100 billion neurons. And so in general, neuroscientists focus on one or a few specific brain regions. One brain region that has been very intensively studied is called the hippocampus. It lies under the cerebral cortex, so you can't see it from the outside of the brain. This is a drawing made around 1903 by a famous neuroscientist, Ramon y Cajal. And what he did, he used a stain developed by an Italian scientist to stain nerve cells in the brain. And then he drew pictures, looked under the microscope and drew what he saw. And in this case, what he did is he cut the hippocampus across its middle. And anyway, Cajal described essentially the cellular architecture of most regions of the brain. So he had an important role in establishing what neurons look like in different brain regions. The hippocampus is very important in learning and memory because information coming from our eyes, our ears, touch, taste, smell, it all funnels into the hippocampus. And this is very important in learning and memory. And I'll talk with my guests about that. So this is a schematic showing this information flow from our sensory organs to regions into the cerebral cortex, visual cortex at the back of the brain, auditory cortex, And of course, these are associated with language areas, right? Speaking, listening, writing, reading. And then other sensory inputs funnel into the hippocampus. Short-term memory occurs there, and then long-term memories are thought to be stored in other brain regions. Also, I mentioned the prefrontal cortex and its role in decision-making. So decisions are made based on information coming in, based on stored information, and making a decision. This slide shows dopaminergic associated circuits. So in gray here are two brain regions, the VTA and the substantia nigra, where dopamine neurons are located. And these dopaminergic neurons play a major role in addiction. They're connected to glutamatergic neurons in these other brain regions. The substantia nigra dopamine neurons, they degenerate in Parkinson's disease. These control motor movements, body movements. And again, they control body movements by affecting both glutamatergic and GABAergic neurons. The brain communicates with the body. One way it does this is through hormones and glands that produce the hormones. So brain cells in a region at the bottom of the brain called the hypothalamus and the pituitary, they produce proteins that are released into the blood and then affect specific organs. So for example, there are hormones, LH, FSH, that affect production of estrogen, testosterone in the sex organs. There's stress hormone, cortisol from the adrenal glands, which I mentioned. That's stimulated by this peptide ACTH. This is what I studied in my master's degree work. And then also growth hormone, which as its name implies, promotes growth, for example, of muscle. And we know how this works in general. The thyroid gland is controlled by the brain. There's hormones produced by the brain that control blood pressure. And then mammary glands, oxytocin. And then not only does the brain communicate with the body, but the body communicates with the brain. One area we'll talk about, which is very interesting, is that the bacteria in the gut can affect the brain. And also the gut can affect the brain by affecting nerves that go from the brain to the gut. And these are in the vagus nerve, which some of you may heard of. This is part of the so-called automatic or autonomic nervous system. So this controls movement of food through the gut. And this is very interesting. For example, people with Parkinson's disease very commonly have problems with constipation, and that has to do with dysfunction of this vagus nerve. But anyway, we'll talk about the bacteria in the gut and the brain, because it turns out that, for example, people with obesity, they have unhealthy bacteria in their gut, and that those unhealthy bacteria in the gut may have adverse effects on the brain. And the good bacteria in the gut may be beneficial for the brain. And I'll talk with experts on how that might occur. I mentioned we'll talk about drugs of abuse, either naturally occurring, morphine, synthetic heroin, fentanyl, the opioids. I mentioned psychedelics, magic mushroom, chemical psilocybin, LSD, DMT. These actually, they act on serotonin receptors and have very interesting effects that we'll talk about. They're not addictive, and turns out a one-time experience with these chemicals can have long-lasting beneficial effects in people with depression and anxiety disorders. And we'll talk about some drugs that activate GABA receptors. These are so-called benzodiazepines. Valium is the classic example. We'll talk about stimulants, cocaine, methamphetamine. These act on proteins that actually remove dopamine, serotonin, and norepinephrine synapses, so they increase the amount there. We'll talk about a couple chemicals that affect glutamate receptors, ketamine, which is very interesting. It has some effects similar to these psychedelics, although it's not as safe, but it will cause hallucinations. And then finally, we're going to talk about neurological disorders. These are self-portraits of a famous Dutch artist. And before he started to develop dementia, and then over about a 4-year period as his dementia progressed. So obviously neuroscientists, neurologists want to know what goes wrong in the brain, how that might be prevented or slowed down in the early stages of the disease. In Alzheimer's disease, the brain simply shrinks, not all of the brain. The frontal cortex shrinks a lot, so this is a normal brain. This is someone with Alzheimer's. The temporal lobe here shrinks. On the other hand, the motor cortex and sensory cortex, occipital cortex, for example, there's very little or no shrinkage. And if one looks at someone in the early stages of dementia by an imaging method that allows one to look at energy metabolism, there's reduced glucose utilization in the brain. So the neurons are not getting enough energy. And we can study not only in humans Alzheimer's disease, we can take after they pass away, can take the brain out and look at what happens. So in case Alzheimer's, there's amyloid plaques and neurofibrillary tangles. And there are also mouse models where mutated genes that cause Alzheimer's disease in in humans, those mutant genes are put into mice, and the mice will develop amyloid. So the green here is amyloid plaques in a mouse brain, an old Alzheimer's mouse. And this is the neurofibrillary tangle type pathology. And these mice will have learning and memory deficits. And we can say, what sort of treatment can we do that might slow this process down? It turns out with these different neurodegenerative disorders, Alzheimer's disease, AD, Parkinson's disease, PD, ALS, Lou Gehrig's disease, most of the cases are what we call sporadic. We don't know the cause. The age of disease onset, that is symptoms appearing, is late, usually after the age of 65, more typically in the 70s and 80s. So that we call late onset Alzheimer's, Parkinson's, ALS. There's a small percentage of cases of AD, PD, and ALS where the person will become symptomatic when they're in their 30s, 40s, or 50s. And these are inherited and caused by gene mutations. And so we'll talk about those inherited diseases and how understanding them may help understand all cases of these diseases. And also for these families that are affected, there are now ways that the disease can be eliminated from the family just over, in theory, one generation through genetic counseling, embryo selection, and in vitro fertilization and embryo selection, so that a woman who has a father or mother with the disease, for example, can ensure that none of their babies will develop these devastating diseases. Okay, this is my last slide. So at this point, I've lined up about 20-some guests that I'll talk with in areas of brain evolution, brain development, what we call neuroplasticity, changes that occur in the brain during adult life. You can look at those there. And we'll talk about body-brain, brain-body, neurological disorders, drugs, therapies, things on the horizon, this new way of manipulating genes called CRISPR, stem cell therapies, for example, either transplanting stem cells into the diseased brain or inducing brain cells, such as glial cells, to form neurons and establish new neural circuits. And that's being worked out now in animals. Brain stimulation, there's already ways, for example, in Parkinson's disease, in people with paralysis, to recover some function. And then finally, we're going to talk about ethics and in genetic engineering and research, in marketing. There's a lot of supplements and so on that you can buy on the internet that the claims made are bogus. For example, improve your learning and memory, and so on. Lobbying, right? Pharmaceutical companies pushing drugs that have minimal or even no beneficial effect. And that's not good. And then disinformation on the internet. That's a big problem that's popped up now with COVID, but it's also true with neuroscience research and brain disorders. So anyway, I hope you're interested in these topics and that you'll join me and my guests, and hopefully I'll have the first podcast up within the next month. I'm recording this just before the holidays, so Merry Christmas, Happy Hanukkah, whatever, and healthy, happy, productive 2022. Goodbye.
Oh, you gotta.